Welcome to our lesson on cloning. The key questions for today are: what is cloning? What are the different processes of cloning? Why is cloning useful? How are adult cells cloned? What are the benefits and risks of cloning? Key words you will need to know are tissue culture, cuttings, identical, embryo, host. Cloning is the process of producing genetically identical organisms from one organism. Both plants and animals can be cloned, but the processes are different. Clones are organisms that are genetically identical. This means they have the same DNA, so would have the same inherited characteristics. Cloning plants has many commercial applications, as a large number of identical plants can be produced very quickly. A desirable parent plant would be selected, commonly after genetic modification or selective breeding, and many copies of that plant are made. There are two main processes that can be done to clone plants. The first is when cuttings are taken from the plant and then cultivated to grow into separate plants. This technique is common for gardeners as it is simple and inexpensive. During this process, a branch is cut from the parent plant and most of the leaves removed. The branch is planted in moist compost or rooting powder, which contains plant hormones, and allowed to grow. The branch will eventually grow roots and become its own individual plant, though it is genetically identical to the plant it was taken from. The second technique is used more on a commercial scale, as it involves the use of some specialist equipment. It does, however, produce many identical plants very quickly. This technique is called a tissue culture. The process involves scraping cells from a leaf of the parent plant and placing the cells in a growth medium such as agar with a mix of plant hormones such as auxin. The cells will grow into tiny plantlets or calices that can then be planted into soil and allowed to grow. Task: the diagram, AQA exam question, shows how new plants are produced using tissue culture. Use the diagram to describe the process of a tissue culture. Some cells are scraped from the parent cell's leaves. These cells are placed in nutrient-rich agar jelly that also contains plant hormones. The cells grow into tiny plants, and once they are large enough, they are planted in soil. Extension: suggest one advantage of using tissue culture and not using cuttings to produce plants. Tissue cultures produce many more offspring than that of a cutting. There is also less damage to the parent plant. Animals can also be cloned to produce several identical organisms. One way this is done is through the splitting and transplantation of an embryo. During this process, an embryo produced by the fertilization of an egg cell with a sperm cell is separated into separate cells. This is done very early on in the embryo's development, so that cell differentiation has not started. The embryo is produced in vitro or in a test tube, so that this can be done quickly. The separate cells then divide to form new embryos, all of which will be identical. These are then implanted into host organisms where they will grow. The offspring produced will all be clones, meaning they are all genetically identical to each other. The offspring are not genetically identical to either of the parent organisms, as they were produced through sexual reproduction, so will have a combination of genes from both parents. The host organism in which it was implanted will not have contributed genetically to the offspring. It will just allow it to develop. Adult cell cloning is when a living organism is cloned to produce a genetically identical copy. The process of adult cell cloning is: the nucleus is removed from an unfertilized egg cell taken from a female organism. This egg cell is now called enucleated. An egg cell is used as it will divide and form an embryo. The nucleus from an adult body cell, such as a skin cell, is inserted into the egg cell. This makes the egg cell include a complete set of chromosomes. The egg cell is then stimulated to divide to form an embryo using electric shocks. These embryo cells contain the same genetic information as the adult skin cell. When the embryo has developed into a large enough ball of cells, it is inserted into the womb of an adult female to grow and develop. This offspring will be genetically identical and therefore a clone of the adult who gave the skin cell. Task: look at the picture below from an AQA exam paper. It shows how mice can be cloned. What colour will the 5 mice offspring be? Why? The mouse offspring will all be black, as they will be clones of the black mouse. This is because the black mouse contributed the DNA, and therefore the genes that were put into the enucleated egg cells. There are often ethical objections to cloning animals, as the general health of the cloned animal is generally poor, and the suffering that could be faced by the animal is unfair. It is currently illegal to clone humans. Extension: answer this AQA exam question. Some children have kidney disease. Kidney disease cannot be cured. In the future, scientists could make a healthy clone of a child with kidney disease. One kidney could then be transplanted from the cloned child into the child with kidney disease. The cloned child would still live with only one remaining kidney. Suggest 2 reasons why people might disagree with cloning a child to get a kidney for transplanting. People could argue that it is unethical, as the rights of the cloned child are not being considered, as it would have to give up a kidney. It would take a long time, and the cloned child would have to be cared for, and the parents may not want to have another child. There are also risks involved during the kidney transplant to both children. It is also illegal to clone humans.